Hi everybody, my name's Sean Perez. I am a PhD student at UC Berkeley studying entomology, which is the study of arthropods or insects. Today I'm here with Oakland Goes Outdoors to do a segment on a Bug Bio BLITZ. So we're going to find lots of bugs and look at their biodiversity, and I'll talk more about that later. We are here at Joaquin Miller Park, which is totally accessible by both car and bus. So get out here with your family and check it out yourself. Before we get started, I just want to mention one really important thing. Right now you see my mask is down, and that's because I don't see anybody within 20 feet of me. So if you do this yourself, just make sure you follow the protocol. If anybody comes within 20 feet or especially 6 feet, throw that mask on. That's what I'll be doing today. Today, we're going to be going over a few things. First, we're going to learn about biodiversity, understand what it is and why it's important. Then, we're going to observe around and we're going to find different habitats and use our understanding of biodiversity to actually measure it in different types of habitats. Lastly, we'll be performing our own experiments by counting up the different types of arthropods we see, and then we're going to compare between these different habitats. We'll explain the results, write it all in our lab notebook. Speaking of which, I want to go over some of the things you will need today. So most importantly, you want to bring yourself a nice comfortable pair of shoes and a mask to be safe. Next, for the experiments, you're definitely going to want a notebook that you're going to keep as your lab notebook. You're going to want a pen or pencil to write things on. And you just want any type of container at home. So here I have two types, just one small Tupperware container and one tiny vial, but anything will work. Okay? And lastly, I just want to note the fact that it's really important to treat all the arthropods we're going to see with respect. So anytime we take an arthropod out from its home and observe it in a vial or something, you want to put it back and make sure the environment is kept the way it is. So with that, let's move on and practice good, healthy science. So what is biodiversity and how do we measure it? So biodiversity can be broken down into two words. Bio, meaning living things, and diversity, meaning variety of things. So you can see biodiversity right here, and it can be a description of different types of animals from, say, owls and wolves. Excellent. So now that we understand what biodiversity is, how do we actually measure it? Well, we can take two steps. First, we pick a group of organisms to study, and then we go out in different environments and we measure the number of types of those organisms we see in each and compare. For this field experiment, we're going to be looking at arthropods. And so how do we describe arthropods? Well, they're animals that have no backbone and an exoskeleton. So sometimes people use the word bug to describe an arthropod. Excellent. So what are the types of arthropods we're going to look for? The first will be insects, which have 6 legs and 3 body parts. And these are probably the most common ones you see. They include butterflies and earwigs. Another kind are the millipedes and centipedes. So these are really elongated and have many legs. It can be a little creepy. Next is the spiders, which have 8 legs and 2 body parts. And I find this spider to be incredibly cute. Then we have crustaceans. You might know these better as pill bugs, the ones we see on land. So these are some of the only ones we will find. So if you see a pill bug, you'll know that's a crustacean. Lastly, we have springtails. These are adorable critters with very cute faces. And you can identify them by their movement oftentimes, and them being sort of like cuddly and small. They take these big jumps, hence the name springtails. Excellent. So now we need to count up the different types of arthropods we see in different environments. So we're going to be looking at 4 environments. The first will be a tree type environment. The second will be a grasslands. The third will be leaf litter. And lastly, we will have a log environment. Excellent. Okay. So we're going to form some hypotheses. We're going to measure the biodiversity of arthropods in each environment. And finally, we're going to compare between them all at the end. See you in the field. After analyzing your results, it's always a good idea to visualize your data in some way. Here, I chose to use a bar chart with environment on the x-axis and the number of arthropod types on the y-axis. You can clearly tell that the grasslands have the lowest biodiversity while the logs have the highest. Visualizing your results helps communicate what you are trying to do to others in a clear and effective way. That wraps up our experiment. I encourage you to go try this yourself, and remember, be safe.